Hello, listeners. I'm really happy sitting down with Francesca Rapino for our Immagina Podcast, where we learn more about the research scientists are doing in the ribosomal translational field. Francesca, could you please introduce yourself and also the work that you're doing at the moment?
Of course. So thank you for having me. I'm Francesca Rapino. I'm a chercheur qualifié in Liège. So I'm a PI of a lab. My topic of research is basically to understand how tRNAs, heterogeneity, and their unknown role in translation. So how the difference in their sequence can impact translation mainly in cancer and specifically in the development or the establishment of cancer stem cells.
And what would be the best outcome of your research? What do you hope to accomplish with what you're discovering at the moment?
So the idea is to actually find, in the best case scenario, signatures of tRNA, so actually variants in the sequence of the tRNA that are important to prime healthy cells, so a subpopulation of healthy cells to actually can sustain the transformation of an oncogenic driving mutation in order to then establish cancer and therefore becoming cancer stem cells. So the idea is to actually try to discover the signature and in the next future, because tRNAs are actually also secreted in the blood, use the signature as a diagnostic marker for example, liquid biopsy that will, of course, improve cancer patient, because by a blood test, you could actually have a marker that would tell you with a certain degree of certitude if you're developing a cancer or not. Of course, then there is also a lot from the molecular biology and translation field. So it's actually not really known how ribosome speed and then therefore protein translation is really optimized. So we have the general routes of regulation, so the main, I would say the main blocks, but it's still not really known how you can fine-tune translation. And of course, understanding how different species of the actors, so tRNAs, tRNA modifications, tRNA fragments can impact on ribosome speed and then on ribosome protein outputs. This is of great interest for, I would say, any kind of fundamental research with any kind afterwards of development into more translational research.
And going back to the specific now, how does RNA phosphorylation, for example, 3-phosphorNA sequencing impact your research?
So when I was a postdoc, we had a lot of problems to actually do RiboSeq nicely, so to really address the speeds of ribosomes and the modification of mRNA impacting on ribosomes speed and protein translation with the old, let's say, kits that were available in the market. There is a lot of steps to do. It's technically very challenging. It's very expensive. And moreover, you need a lot of material to get results that you can actually trust. So basically, when I first got in touch with Immagina was because the products that they were developing were much more related to a more feasible research. So actually a kind of less time consuming, much less material, which of course for people like me that work with stem cells, this is key because we go on tissues. And what we actually are interested in is in the active ribosome translation. This kind of products for researchers like us will speed up and make much more routine and less costly the research we are doing. So now we are a bit bound to the fact that the majority of the experiments we cannot actually do or not do it nicely because there are not technical supports to help us to really look deeply into tissue. We normally do this in cell lines. In yeast, there is a lot of research because you can, grow them like crazy. Now, the output that I hope to achieve is to go on a really more clinical tissue level to really follow ribosome speed, ribosome translation, and all the different aspects of translation through those technological supports.
A good question to tie it all in right now is, can you describe how Immagina Biotech could help your science?
It's been a very nice relationship up to now. We started collaborating not so long ago. What I really like is that there is a lot of honesty in describing what the technological support can do and cannot do. So now, for example, we're doing our first setup on stem cells from intestinal murine system. So really, really few material. It's 3% of your intestine. And we had, of course, a lot of technological challenges, and the supports in Immagina were really helpful setting up this technology, which now is running with a tissue that was not into the set of the kids. So I think the good thing is not only to have technology, but also to have people that know this technology. They're not part of a huge company and they've never been into a lab, but they are actually into the field and they can really do a kind of personalized support to us. And this for me is the most valuable thing, because good technology many companies have. The problem is that if the good technology is not feasible for what you're doing, that good technology is useless to you. And so for us, the technical support has been really the added value, I would say, to our relationship with Immagina today.
Francesca, that was I'm really in awe of your research and also the energy that you bring. So thank you so much for sitting down with me.